Okay, can you hear me now? Yeah, brilliant. Okay, so I think that's generated some really interesting discussions. And while we wait for questions in the room and online, I had an initial question, and I think Rena, you really made me think. I thought it was a great idea, the fact that now we're moving testing and molecular testing to sort of, not by product, but more by class of products. And I think, that's going to, I think, really help in new research, because you're absolutely right. If you have a drug target and you have a mutation and you've got 5 different tests that show you the mutation, it's a very binary. I get it when there's maybe different levels of something that maybe is more drug specific. But I was wondering, maybe Olga, you can invite. Are we doing something similar in the EU? Are we going to do some sort of? Is that available for us here? Sort of looking at maybe CE mark tests or in vitro diagnostic tests that are maybe applicable to a group of products or just to a mutation itself, rather than linking to medicines. And then maybe we'll pass to Zahar how that is linked to reimbursement as well.
Yeah, I would say it's more related to the topic of tomorrow, but I can maybe already comment that in the IVDR, the IVDs or companion diagnostics are linked to the INN, but it potentially could be linked to different INN as well. So it's more a question of discussion in the relevant forums with the companion diagnostic and IVD maybe related groups.
So you're saying it doesn't necessarily need to be linked to the product. There's maybe a scope to link it to a mutation instead.
No, it is linked to the specific product. It should be there in the label.
Okay.
But
okay, so we haven't quite got there, but maybe that is a challenge to try and make these more accessible. And is there anything from a payer's perspective in terms of how tests are used? Would you reimburse a product that maybe hasn't got a test approved? Let's say you've managed to get local tests and they do it locally, and maybe there isn't a centralized CE mark test. Would that affect how you would reimburse?
So the question is whether we want to go to where FDA is now, right now, having certain companion diagnostics. There are some advantages, but I also see some disadvantages, because then you're tied to certain technology, to certain vendor, and it also makes us not really flexible. It also makes it difficult to innovate, to find better tests, to experiment. So in the Netherlands, currently we have, the centers can develop their own tests and use their own tests. I'm not sure how the situation would be in the future, whether we need to use certain tests. One of examples, one of our problems right now is that we don't have any test that can define HRD, because there are several tests available, but none of them seems to be really good. Certainly not the one that has been, I guess, FDA approved. There is this one Myriad test, I guess. Several centers in the Netherlands have tested, are using that test and comparing that with other tests, and they see many false positive or negatives. Not sure, but it's not really reliable. So choosing the test is difficult, but currently the hospitals can choose themselves, can choose which test they're going to use, which is also making things difficult, because then as long as we don't have any threshold, as long as we don't have any reliable tests, then if you get tested in one hospital, you can be positive and get the drug, and then if you're tested in another hospital, you can be negative and don't get the drug. So we need more harmonization, and I hope that in the future we can have more harmonization for different tests, allowing new tests to be developed, but at the same time helping us to innovate.
Thank you. While we wait for colleagues in the room, I've got a question here on Slido for you, Sahar. They say, this question is anonymous. It comes, with the increasing use of biomarker to limit reimbursement, don't we risk undertreating certain groups of patients? Example, from HER2 to HER2 low, ultra low.
The HER2 low, yeah, I know what you mean. Yes, I guess when the drugs, just before they are entering the market, we need to develop the tests. We need to implement the tests. So when the drug is launched in each country, then we have a reliable test, and perhaps use this moment to have more centralized testing to reduce the practice variation. Because if each hospital needs to validate a new test, then it would be very costly, and it would probably takes a long time before all the centers have implemented testing. So I guess you need to have several centers who can do the testing properly and then launch the medicine.
All right, thank you. All right, I think we've got a question from the colleague in the room.
Mark Lawler, Queen's University Belfast and CDDF. Sahar, I really like the Dutch method, the approach that you've used. How do we get the rest of Europe to adopt it?
You can use me as ambassador.
I think you make a very good ambassador.
Which part of it did you like? Because it was just like about all our uncertainties. So what part did you like?
Well, I just think the approach you've used, I know there are uncertainties associated, but I just think the approach you use, you've involved patients, you've brought everybody together. I think as well, I mean, I'm obviously very familiar with the DRUP trial, which is something slightly different, but I have a sort of a follow-up question about that. But I just think the approach in terms of thinking rationally and pragmatically about how you deliver to patients is actually something that if some of our bodies in our own countries thought about it more, would be really attractive.
Yes, absolutely. We have had a very positive experience with including patients in our decisions. And they are very knowledgeable, and they decide together with us. So I would really recommend to have the same tripartite committees in your countries, and just, I mean, you can discuss all the uncertainties together, and after a while, everybody actually can decide on these difficult matters.
And second question then was, I've always thought the DRUP approach is, very, again, practical and probably better than the US Genie approach. But what challenges have some of the other countries faced in taking on a DRUP or a DRUP-like approach?
So the major challenge is first testing, because you are now testing for screening for inclusion within a clinical trial. The patients that are getting access to the DRUP trial are the patients who have exhausted all the regular treatment options. So at this stage, usually there is no reimbursement for testing. This is one of the challenges. The other challenge is having all the drugs available within your platform. The DRUP trial currently, as I said, has got like 37 drugs. So I'm very happy that companies are working with us. But one of the reasons that they work with us is that they know that there eventually would be a reimbursement in a third stage. Because if you just stop after first and second stage, or after third stage, and then again, there is no reimbursement for the rest of the patients outside of the trial, then it's not, there is no incentive for companies to make the drugs available. And the incentive for us as payers, as health system, is that then, I mean, before DRUP trial, before having DRUP trial implemented in our health system, we would receive many off-label many hospitals would ask for off-label reimbursement, which is not helping because you're not gathering data. Yeah. So eventually we wouldn't know whether the off-label use of those drugs are really working. And another thing would be that access would be very much depend on your health insurance company, whether they are willing to reimburse or not. So this is the reason that we decided we need to embrace DRUP trial and have a smooth pathway from the experimental part to the reimbursement parts, because these are really rare indications, and it would be fair to give those patients access to the treatment as well.
Yeah, and I think the key thing is you're collecting data continuously as you go along, which I think is really important.
Exactly.
Thank you very much.
Exactly. Thank you.
Okay, I think one from Jan.
Yeah, I'd like to echo what Mark said, and as a Dutch, I'm proud of the fact that our payers taken a proactive approach in this and not simply denied access to drugs. Yet there's another side of that coin, of course, there always is. And I understand that nivolumab is now available for MSI high, just like pembrolizumab was already available and is FDA approval, I know of that. I don't know the data for the nivolumab study, but I do know the data for the pembrolizumab study. And what I have always been fascinated with, if you take the totality of the patients in that trial, you can understand that MSI high leads to approval. I used to be working in sarcoma. There were 9 patients with sarcomas in that trial. None of them responded. So is that the population that we really should approve a drug for use on if the data show that there's 0 out of 9 responses? Because the totality of the study gives you another picture.
Maybe should we pass on maybe to Ola?
Yeah, maybe you can say, but I guess there are responses in the trial, so I'm not sure.
I think with sarcoma there were none, but
with sarcoma,
yes, there were responses. I'm sure.
I understand.
But you see, there's breast cancer, non-small cell lung cancer, whatever, MSI high, no doubt, because the numbers are big, and there's proof there. But there were other entities which were small numbers of patients, 6, 9, 3 maybe, not a single response there. And then the question is, should we have approved the drug for use in those populations as well? I'm not giving you an answer because I don't know. I'm just asking the question.
I understand. So that's what I liked in your slide was this indication with let's see. It was first the label was for exon 19 and exon 21, and then you said for non-resistant ones. So is anything like tumor agnostic? Does it really exist? So perhaps there are always some patients who would never respond to such drugs.
And maybe that sort of question then for the regulators is, for such indications like EGFR non-resistant, sort of how much evidence or what's the level of evidence do you need to get to that point? Did you have to get every single possible mutation clinical data, or do you
so how much that example, there is no clinical evidence for that. It's more mechanistic and non-clinical evidence in silico models and cell line studies. So that's for a very rare mutations when you can't get clinical samples to study. So that's so it's not like for something that you will be able to study. And we don't have that many examples. It started with actually when cystic fibrosis, that was the first example, and then the second one was just the EGFR. We haven't had many more examples for that, for following that guidance. In oncology, I'm only aware of this EGFR example. But I guess your question is more of for the tumor type. And we have had this internal discussions also within the office. So right now, it is approved in all tumor types that meet this criteria, like if it's a biomarker positive, they are eligible, but at the same time, it's all clearly laid out in the label. So oncologists whether they look at it or not, but that's, it's hard to actually then say, oh, it doesn't work just in this, or it doesn't work, What if maybe?
No, but you have to set some limits somewhere.
Right.
Nobody knows the answer to what it is.
We have had. So it has to work at least. You'll see it has to work at least some around 10 tumor types before you say it's tissue agnostic. So it's not like if it's 1 or 2 or maybe 4. So there is a number that the review team looks into, whether does it work in this many tumor types.
But then the evidence that comes from the TRK inhibitors that's undebatable, I guess, because throughout tumor types the response is the same basically, as long as the target is there. For the MSI high, there is this imbalance because there are patient populations that do wonderful on this, and others that have been studied to a limited extent. I understand that the minimum number that you need for approval is something like 25 or so in a patient, in a specific tumor with this particular
unless you
target.
You will say it's approved only in these tumor types, you'll have to spell it out.
I think that's what.
Whether we'll go there, I don't know, but that's that's got a lot of internal debate on this topic also, whether it should be just tumor agnostic versus. But this again. whether that 4 patients or whatever, are they representative of the real population?
Nobody knows, but if you have 0 out of 4
Yeah.
as non-evidence,
Yeah,
then still
exactly.
the question,
Yeah.
you would have 1 out of 4.
This,
Okay.
Yeah, this was actually a lot of discussed during the TMB high, because there were clearly there was one tumor type that wasn't showing any. So maybe there may be when full approval comes on board, whether there will be a restriction, we don't know.
Yeah. Thanks.
But it's an interesting debate, isn't it? Because ultimately, for these tumor agnostic, these non-resistant type things, they're for last line, and yeah, when you start getting to 4 patients, 3 patients, I mean, is that really going to be enough to be conclusive? I think if you have a strong mechanism of action and these patients don't have anything, I do see why there's a positive benefit risk there. Because otherwise, what can this patient has, and there's enough there, But maybe, Olga, do you want to comment on the tumor agnostic, non-resistant type?
Yeah, I will try.
Space.
So, but as a general case, not for specific cases. So I would say, as you said, it depends on the mechanism of action. And actually, as I also explained, what you expect as a context dependency. So whether it would be a major proliferative alteration that really in each and every cancer results in the hyperproliferative phenotype, then you would expect it working across all the tumor types. But for some mechanisms of action, it's becoming more difficult, like immune-related mechanisms of action, you have very high variability between patients, and even with targeting specific alterations, you may not target the majority of patients.
And I guess that's why we've seen this limited to more rarer types. And I guess we've seen how when we try to do this for FGFR3s and it becomes a much more bigger sample set, we're starting to have difficulties in terms of that. Interesting. So I'm just looking here for Slido. So we've got Dr. Phillips, we've got another question for you, quite specific but interesting. When developing a liquid and tissue NGS code in parallel, do both need to be approved contemporaneously with the drug?
Yeah, the answer is yes. If both liquid and tissue were used for a clinical trial enrollment, they have to develop both to bring in as a companion diagnostic for both tissue and liquid. But sometimes, usually, it's only enrolled using tissue, and the liquid is just collected along the side. So that's so they are only bringing in tissue, and then the liquid is brought in later. Just like the EGFR example, EGFR was enrolled only based on tissue. Yeah.
And maybe the same question for the EMA, would that matter, or would it also depend on how they've done the trial?
Well, there is no approval of the companion diagnostic at the same time of medicinal product. What we encourage is co-development and the availability of companion diagnostic simultaneously with the medicinal product to ensure access to patients to both. But when you consider as different types of tests, this is coming back to the concept of homogeneity that I explained. To what extent you would expect the homogeneous treatment effects depending on the different tests.
Okay, I think we've got a colleague in the room.
Yeah, thank you. Claudia Pop from Roche. Thank you so much for these interesting talks. I think it shows really nicely the complexity. I have a question with regard to the pilot for in-house tests or lab-developed tests in US. I think it's an interesting approach, but I noticed also that end of September, FDA published some proposal for some stricter rules with regard to lab-developed tests. So is it not a bit contradictory? Because, I would be interested to hear if, in Europe we have already now some rules for lab-developed tests within the IVDR. What is the perspective in US? Will you go this in a similar way, in similar direction? If you could say a few words, would be great.
Yeah, so I'm not sure whether it's contradictory, but it's... maybe I didn't get a lot of time to talk about the pilot, but that's I guess in the previous conversations also, we did hear how the local developed tests are used, and some of them are not really validated. And so how can you ensure the patients get the right tests. So this pilot is an approach towards making the locally developed tests more consistent, and the performance characteristics of the tests are, are developed in using the minimum performance characteristics that will be laid out along with FDA when a drug is approved. And we are hoping that will help improve patient care by using local tests that will meet this minimum performance characteristics, even if they are going to be developed after the drug is approved. Like, let's say, the goal is when pharma companies are using tests for enrollment into the clinical trial, they will be actually enrolling local tests that meet minimum performance characteristics. So when they are bringing in the therapeutic drug to FDA for approval, they will actually, list this was the minimum performance characteristics of the tests that were used for enrollment. And so that's what is going to go into the FDA website. And then future local tests who are going to add this biomarker into their panel, they will make sure their testing will have this minimum performance characteristics. And also, I think I was hearing some of the the earlier conversations about how we do not mandate central testing for all clinical trials prior to enrollment, because we understand it's the difficulty, that especially with regards to the rare biomarkers. Only if the trial is significant risk trial. And that's again, I didn't get a lot of time to go through significant versus non-significant. A lot of trials are not significant risk trials. Only if they meet that specific criteria, they are significant risk trials, and then they need an IDE. But when they need an IDE, of course, then you have to have at least pick one or two, whatever tests that you are planning to enroll. But if majority of the trials done are non-significant risk trials, which means they are maybe enrolled using local tests. And but the hope is this minimum performance characteristics will improve that to make sure that the clinical trials will be enrolled using good tests that meet minimum performance. And then downstream also, when it's used in patient care, they will be using local tests that will have minimum performance characteristics.
I have a question for actually both of you. So how easy should it be to implement the learnings from real world? So this year there was a publication, a joint publication by Netherlands and UK, showing that patients with G12 RAS mutations that they do not respond to certain chemotherapy, to Lonsurf, so we discussed that with EMA, and EMA said as long as the company doesn't collaborate, we are not going to do anything with the data. How is the situation in FDA, and how do you think how easy should it be to implement the learnings from real world data to actually optimize patients' treatment?
Sure, I think that's a very tough question. That we do run into that, but the good thing is, I guess, NCCN updates gets so there are some biomarkers that are maybe FDA didn't approve it for that particular reason, but the intended use, but it gets into NCCN and the oncologist uses it. So I guess sometimes they work with the NCCN in order to get that into the guidelines. And I mean, there are ways to bring, because then it has to be done by some specific diagnostic company, because if it's a chemo, there is no drug labeling change or anything. But there is a way to get these biomarkers approved. I mean, if it's a PMA or 510, depending on. But it usually, I mean, I haven't seen any examples, but we have discussed a lot on it. There are some companies interested in and how they will bring it forward. We have thought of, some sort of guidances how to, like use of biomarkers that do not reference a drug. There should be a mechanism to do it, but we haven't done it. But I guess how it's worked is by getting into the guidelines.
So yes, in Europe, ESMO has also implemented it. What do you think? Should it be something that the label, the text of the authorized text, should it be adapted based on the learnings of post-marketing learnings, or should we just stick to the clinical trial and what was done there?
Well, I do not know the specific case, but what I think about, it's about extrapolation, what you could extrapolate based on mechanism of action and whether you need there to confirm this effect by clinical data, and then what level of evidence you would need in this case, whether you would need the level of evidence of clinical trials or real world data, and what is the quality of the data.
Yes, I understand. Yeah, this was something that has been tested in 3 different cohorts. But yes, it's usually the label is updated based on safety, but not this kind of molecular data. And maybe in the future we should move to implementing the learnings that we get from sequencing.
Yeah, but we have to be careful with sequencing, because you also touched on something, and it made me think. You said, "Oh, so we need to maybe sequence this medicine second line because the overall survival from real world data, or whatever, doesn't change." But I would argue, if you've got the best medicine and you're the patient, first line, you want the best medicine. Yeah, I know I might want to save it to get the second line, but the reality is, some people don't get there. So I think we have to be careful sometimes with these real world evidence and things, and maybe overall survival doesn't, but maybe to that one patient, it might matter that they get given the best treatment.
Yes, sure. And
if you have something that,
I agree, but if you have found a biomarker of non-response,
of course.
So and those information are not included now within the SPC in Europe, and I guess it should be, because then you actually the guidelines are okay, but not every insurance company covers everything in the guideline, but they would cover if it's asked by EMA regulatory agencies.
Okay, I think we're just coming to time, but I think that's Ruth, but I can't see all the lights. But why don't you go ahead and ask questions?
Well, it was really a follow-up on that, because it's a fascinating bit of data, because basically what they said is there are patients who don't respond, who are really unlikely to respond. And Lonsurf is actually, speaking of somebody who treats patients with it, it's not that well tolerated, and patients feel pretty rubbish on it. So how do we handle that sort of post-marketing? And maybe it's not the regulators. Should this not be the payers? But then how do you regulate the collection of real-world data? Now, I guess we're actually going to have a session in that in the annual meeting of CDDF, as this starts to come out. But it was really a question to Rena in particular. In terms of real-world data, how is that going to be regulated? Because it's very powerful data, but it's very different from a clinical trial.
Yeah, I mean, there have been, I think there's one approval where the real-world data was included as a supporting evidence, but it's got a lot of weight, how there's a lot of caveat in how you use that, and because there's a lot of bias that can be associated with what you get as real-world data. So I'll be very careful, in saying that, real-world data can be used for regulatory decision-making, because it's because of all the, details associated with using real-world data. And there is a good group working on it. There are some discussions on, especially for the recent one of the ODAC said, if anyone was listening, for the control group, how the real-world can be used. And so there have been some applications where people are looking into using real-world data, but the robust evidence that's needed is very important.
Right, thank you. I think, Olga, would you like to quickly comment?
I will just comment that it's used, but for contextualizing the data.
Okay. All right, I think we're on top of time. We have so many questions as well. It's been a really good topic. I want to thank all the speakers and for this great panel discussion. I think it's been a great session. So thank you, everyone. I really appreciate it. And for the questions as well.
Thanks to you, Richard.
Thank you.
Thanks.